Hi everyone, this is part 2 of chapter 14, Transcription and RNA Processing. There's a distinct difference in the process of transcription, making RNA, between prokaryotes and eukaryotes. In prokaryotes, all of transcription, DNA replication, and protein synthesis is contained within a single compartment. Remember, prokaryotic cells have no nuclei. In eukaryotic cells, however, because there is a nuclei, there's a compartmentalization of the process of transcription and translation. And over the course of evolution, that's had distinct consequences for the processing of RNA and the modification of RNA molecules after they have been transcribed. The basic process of transcription is similar. However, what happens to the RNA during transcription and after transcription is a little bit different in eukaryotes versus prokaryotes. In eukaryotes, in addition to the process of transcription, we are going to talk about processing, and then later on in the chapter 15, we're going to talk about the process of translation. The structure of the RNA polymerase from bacteria and eukaryotes is quite similar. This is this clam-shaped molecule. The one difference is that eukaryotic polymerases are bigger than prokaryotic polymerases, and there are 3 distinct types of RNA polymerases in prokaryotes, whereas E. coli and other prokaryotes have a single RNA polymerase, but they do switch out different sigma factors. So nuclear DNA is transcribed by 3 different RNA polymerases in eukaryotes. There's RNA polymerase 1, which transcribes all of the ribosomal RNA genes. There is RNA polymerase 2 that transcribes all the structural genes, the messenger RNA genes, the messenger RNAs that make proteins. And RNA polymerase 3 takes care of the tRNA genes and some other small RNA genes as well. Okay, so here's our RNA polymerase, just like we saw in prokaryotes. It has the thumb and the palm. There's a few more features that are described here. The DNA comes in through this pore right here. It's clamped onto by this portion of the RNA polymerase. It has a pore for the NTPs, not the deoxynucleotide triphosphates, but just regular nucleotide triphosphates, because we're making RNA here. This is where the DNA is coming in, and this is where it's leaving up here. This is the exit, this is the entrance. And here's our RNA, it's exiting the same spot that the DNA does. There's a very important factor that has to be bound to all polymerases. Magnesium is a very, very important cofactor for the formation of the phosphodiester bonds and binding of the nucleotides as they come in. And if you take molecular biology, we'll discuss that in some more detail. Okay, so how does the promoter region of a eukaryotic gene differ from a prokaryotic gene? They have similar organization of elements, but there are distinct differences. So the core promoter has in it a Tata box, which is similar to the -10 region, and it also has a lot of other upstream sequences that help to regulate the binding of RNA polymerase at the core promoter. Here's where the transcriptional start site is. So if you remember in prokaryotic promoters, this AT-rich region was approximately -10 nucleotides up from the start site. Here it's about 25 nucleotides away from the start site of transcription. This is what the Tata box, this is the consensus sequence for the Tata box. It's very similar to the prokaryotic sequence. There's a transcriptional start site, and this is the consensus sequence here where you have 2 pyrimidines, a C, an A, and then 5 pyrimidines in a row with surrounding the transcriptional start site. And many factors bind in this region of -10 to a -100. This is some regulatory elements are here, some common regulatory elements are here, and then there are many other sites for specific regulatory proteins to bind to elements in the DNA. They can be close to the start site and they can be very far away, thousands of nucleotides away. Okay, all of these sequences bind transcription factors, which if you take cell biology and molecular biology and biotechnology, you will find out a lot more about them. We're going to talk a little bit more about that later on in the semester when we talk about genes that are expressed during embryonic development. But transcription factors are very important in regulating whether or not a gene produces a messenger RNA, is turned on or turned off. Okay, we're going to focus most of our attention on RNA polymerase II because that is the RNA polymerase II. That is the polymerase that transcribes genes into messenger RNAs that get translated into protein. So the first thing we're going to look at is the Tata box. All polymerase II genes have a Tata box. And to this Tata box, one of the first factors that binds is TF2D. TF2D is actually a large complex of proteins. It has many subunits to it. One of the subunits is the actual Tata binding protein, the protein that actually contacts the DNA. An interesting thing about Tata binding protein is that it is one of the few proteins that binds to DNA in the minor groove. Okay, so TF2D binds to the Tata box. TF2D is a complex of proteins that includes the Tata binding protein and several Tata binding associated factors, or TAFs. So you have Tata binding proteins and Tata binding protein associated factors, and they all make up TF2D. TF2B binds to TF2D. TF2B acts as a bridge to bind RNA polymerase II and TF2F. Now you don't have to remember all of these different proteins and complexes. You should remember the Tata binding protein and the Tata binding protein associated factors because those are the first ones that bind. All the rest of the proteins that bind to this, if you notice, they are bending the DNA, and we know what happens to AT-rich regions of a DNA molecule when the DNA is bent. They tend to break their hydrogen bonds and pop open. So once TF2B and TF2F bind, they recruit or assist in the binding of RNA polymerase II. Then a few more factors, E and H, bind to RNA polymerase II, and they form a pre-initiation or closed complex. So this is the same idea as in prokaryotic transcription. We're starting out with a closed complex that's bound tightly at the promoter region. TF2H acts as a helicase to form an open complex. So we need something to help to stretch the two strands apart, break these hydrogen bonds, and TF2H is just that protein. TF2H also has a very important function in that it phosphorylates the C-terminal domain of RNA polymerase II. Okay? RNA polymerase II, eukaryotic RNA polymerase II, has a very important domain called the C-terminal domain. A lot of important functions happen in the C-terminal domain of RNA polymerase II, and we're going to come back to that towards the end of this lecture. Once the C-terminal domain is phosphorylated, this breaks the contact between TF2B and RNA polymerase II. All of these factors are released and you transition from a closed to an open complex. Okay? The C-terminal domain of RNA polymerase II, we'll come back to this again. Okay? So the important thing to remember about the C-terminal domain right now is that there's a lot of different functions that are controlled by the C-terminal domain, and in order to switch those functions, you phosphorylate or remove the phosphate groups from the C-terminal domain. So we have a stretch of amino acids here that bind phosphates or release phosphates, and the binding and the release of those phosphates change the function of the C-terminal domain at different points in the process of transcription. So we transition from a closed to an open complex. All those factors are released after the open complex is formed. Very similar to prokaryotic transcription when we release sigma factor in the transition from a closed to an open complex. Okay, so next is the elongation phase. Just wanted to show you that there are lots of different subunits of proteins involved in all of these different factors. Here's TF2D, for example, with TATA binding protein and all of these TATA binding protein associated factors. How many are there? Well, 10 of them. RNA polymerase II itself has many different subunits to it. These are TF2B, just one, TF2F, three, TF2E, 2, TF2H, a whole bunch. So it's just important to remember that when we're talking about these different factors, that they're actually comprised of many different subunits. Each of these represents, of course, a single polypeptide. Okay, so we talked about initiation. We mentioned the elongation phase. We're going to talk a lot more about the elongation phase and some of the processing steps that happen in it. But in termination in eukaryotic transcription is a little fuzzy. Okay, there's three types of transcripts that terminate differently. tRNA and ribosomal RNA have specific functions that can terminate transcription. They are more like prokaryotic transcription termination sequences. However, Pol2 transcripts, remember RNA polymerase II is the polymerase that transcribes messenger RNA. So Pol2 transcripts terminate when the 3' end is cut of the messenger RNA itself is cut just past the polyadenylation signal sequence. We're going to talk about what this is, polyadenylation sequence. Okay, so in Pol2 transcripts, the RNA is kind of cut off, kind of like you'd cut off a strand of Play-Doh with a plastic knife. When you release the RNA from the complex, that when it's bound to the DNA at the transcription bubble, then the RNA polymerase then just falls off. It stops transcribing. Okay, because of that compartmentalization of RNA, having the necessity of making sure RNA remains intact while it is being processed in the nucleus and transported out of the nucleus into the cytoplasm and then translated, there are a lot of modifications that have occur over the course of evolution to make sure that messenger RNA remains intact. tRNAs, ribosomal RNAs, and messenger RNAs are all modified. Ribosomal RNAs have spacer regions removed. tRNAs are usually not spliced, but they are trimmed by exonucleases that chew away some of the nucleotides at the ends, at the 5' and the 3' ends. They also have some modifications made to their nucleotides. So sometimes unusual bases are substituted in for different tRNA molecules. In messenger RNAs, the ones we're going to pay most attention to are capped at their 5' end. They have a special modification at their 5' end. They are spliced to remove introns, and they are polyadenylated at their 3' ends, also to protect the messenger RNA from being degraded. So let's look at a ribosomal RNA gene first. Ribosomes have different RNAs that make up the small subunit of the ribosome and the large subunit of the ribosome. This also goes in the large subunit of the ribosome. And these RNAs are transcribed as a single transcription unit, okay, which is subsequently cleaved into smaller RNAs. So here's the DNA. This is what gets transcribed. Okay, so RNA polymerase binds here, RNA polymerase 1 binds here, makes a copy of all of this DNA here, and then the RNA is chopped up into the correct pieces, and then these RNAs then form the ribosome. The processing occurs in the nucleolus. Okay, that's a section of the nucleus where the RNA for a ribosome is chopped into the correct pieces, and ribosomes are actually assembled in the nucleus, inside the nucleus, before they get transported out of the nucleus and into the cytoplasm. These are the 3 functional RNAs that are important to ribosome structure and function. Okay, what about messenger RNA processing? Before it leaves the nucleus, eukaryotic messenger RNA is spliced, capped at the 5' end, and a PolyA tail is added at the 3' end. This diagram shows a very interesting phenomenon called self-splicing. There are some eukaryotic organisms that splice out their own introns. Okay, there's a group 1 splicing mechanism and a group 2 splicing mechanism. Both of these exist in eukaryotes. This group 2 splicing mechanism is very similar to what goes on in most eukaryotic messenger RNAs in higher eukaryotes. So one of the first things that happens in the splicing reaction is that a central adenine attacks a nucleotide over here and makes what's called a lariat structure. Okay, so there's actually a circular structure here where this adenine has its normal 5', 3' phosphodiester bond, but it also has its 2' carbon associated with another nucleotide. Okay. Once that happens, this 3' end attacks the 3' end of the intron here, and the whole intron is released. And this can happen all by itself without any associated proteins in certain single-celled organisms, eukaryotes though, nonetheless. In higher eukaryotic organisms, the same chemistry happens, but in order for it to proceed in an ordered and regulated fashion, there is a complex of many different proteins called the spliceosome that accomplishes this. So the same chemistry of this 2' attack on the 5' end occurs to make this 2' phosphodiester bond, and then this 3' end of the exon here attacks the end of the intron, and you've removed your intron here, in this lariat structure where you have a loop and a tail. To break this down a little bit more, introns are the regions that have to be removed from messenger RNAs. You need to hook up an exon 1 here with an exon 2 in a very precise linkage so that you do not introduce any stray nucleotides or remove any nucleotides by accident at either end. It has to be a very precise splicing. All introns have at their 5' end a GU and at their 3' end an AG sequence at the very end. It's very important to remember that this sequence, GU-AG, is at every single intron that's ever been discovered in all eukaryotes. Okay, so the first step in the splicing is binding of components of the spliceosome at the branch site, which is always an adenine residue in the middle of the exon. U1 is a complex that has an RNA and protein in it and binds to the 5' splice site right over here. And then U2 binds to the branch site. U1 snRNP, it is a small nuclear ribonuclear protein particle. That means it has proteins, peptides, polypeptides, plus a small RNA that's bound in a complex here. And it recognizes the 5' splice site. U2 binds to the branch site. U4 and U6 and U5 trimer, all three of these subunits bind together. The intron loops out and the exons are brought close together. Okay. There's our branch site, the A residue. That's U1, U2, U4, U6, U5. And then the 5' splice site is cut, and the 5' end of the intron is connected at the branch site right here to form a lariat. And then U1 and U4 are released. Those complexes are released. The 3' splice site is cut. Exon 1 is connected to exon 2. This is a simultaneous reaction. It's not cut first and then added. The cutting and the adding happens at the same time. And eventually, the intron will be degraded, and you have the intron. This will float away. It lasts for a little while. You can measure it, but it eventually gets degraded. And now you have your exon 1 and your exon 2 spliced together without any extraneous RNA sequence in the middle. And when you go to translate this, you'll get a seamless sequence of nucleotides to make your polypeptide. One very interesting feature of the human genome, or a higher eukaryotic genome, is that we can produce many more polypeptides per genome than we have genes for. And one of the ways that that's accomplished is by alternative splicing. Alternative splicing means that a gene can have different alternate exons within a gene structure, and depending upon the tissue or the environmental conditions or the microenvironment that the cell finds itself in, you can choose between alternatives of a single exon to put it in the final protein product. This generates great diversity and allows humans to make 4 times as many proteins as they have genes for. So here's an example of a eukaryotic gene. The gene is for Alphatropomyosin. It's a gene that's found in muscle tissue. And it can make 9 different protein products from the same gene. Okay? This is the one that's found in striated muscle. This is smooth muscle, brain, fibroblasts, etc., etc. How do you make 9 different proteins from a single gene? So here is the primary transcript. In all of these tissues, this is what's first produced, the same primary transcript. But during the process of splicing, messenger RNA splicing, we're going to pick and choose different exons to include in the final messenger RNA and remove the introns that we don't want at all. The introns are these light green regions in between the more brightly colored exons. So let's look at what happens in striated and smooth muscle. Okay, both striated and smooth muscle retain exon 1, but as far as exon 2A and 2B is concerned, striated muscle chooses exon 2A, whereas 2B, excuse me, and smooth muscle chooses exon 2A. Okay, what about this 1B exon? Neither one of these transcripts includes 1B. So during the splicing process, we splice out this region here for striated muscle, this region here for striated muscle. For striated muscle, we remove this region here. We keep exons 3, 4, and 5. If you notice, all of the proteins keep exons 3, 4, and 5. And for striated muscle, also we're going to skip over exon 6A, keep exon 6B. We'll keep 7 and 8. We'll keep 9A and 9B. And that's it, but not 9C or D. Okay, that's the end of the protein for the striated muscle version of alpha-tropomyosin. The smooth muscle version, okay, we skip over exon 2B and 1B. Okay, so when we splice out the messenger RNA, we splice all of this away. We keep exons 3, 4, 5, not 6A, 6B, 7, 7A, and we'll skip over exons 9A, 9B, 9C, and just include 9D. So in the primary transcript for the smooth muscle, we're going to splice out this whole section of the primary transcript and hook up this exon with the end of this exon over here. This, of course, is the reaction that occurs during the splicing process. One of the most important modifications is the capping reaction. Okay, so here we have our messenger RNA. It's got a 3' end and it's got a 5' end. You notice that the 5' end here has its typical triphosphate here. This is uncapped. In the capping process, we're going to modify what's attached here. So this is the rest of the messenger RNA that goes down here. The first thing that happens is that the phosphate is removed. Okay. And then an enzyme called guanylyl transferase attaches GMP to the 5' end. Okay, by hydrolyzing the pyrophosphate that's attached to this GTP. So if you cut the nucleotide right here, this would have had 2 more phosphates on it. You cleave those 2 off as pyrophosphate, and now this phosphate came in with the GTP, and these are the 2 phosphates that were remaining before. So now you've got a guanylyl GTP attached to the 5' carbon with a triphosphate in between. To make it a little more stable, we'll add a methyl group here, and that's the final structure of the 7-methylguanosine cap. Okay, this is it. So it's a 5' 5' carbon. Okay, what about the other end of the molecule, the PolyA tail? Every messenger RNA has a polyadenylation signal sequence towards the end of the transcript. AAU, AAA, that's pretty universal. What happens when this is transcribed is that an endonuclease cleaves the messenger RNA away from the DNA that it's attached to at about 20 nucleotides downstream from this cleavage and polyadenylation signal. Okay, and then an enzyme called PolyA polymerase comes in and attaches about 200 adenosine residues at the end. Okay, so there's a PolyA tail that protects the 3' end of the messenger RNA. So now we have a messenger RNA that is protected at the 5' end with a 7-methylguanosine cap and at the 3' end with a PolyA tail. How does this all happen during the process of transcription? Well, remember I told you to pay attention to the C-terminal domain of RNA polymerase II? Okay, here's RNA polymerase II, and here's its C-terminal domain. It's the last little bit that sticks out away from RNA polymerase II. And see all these little P's here? This indicates amino acids that could be phosphorylated. Okay. The thing to remember about the difference between capping and splicing and cleavage and polyadenylation is that the pattern of phosphorylation that occurs on this C-terminal domain is different for the different functions. Okay? So when the C-terminal domain is regulating 5' end capping, it has one pattern of phosphorylation. When the C-terminal domain is regulating RNA splicing, it has a different pattern of phosphorylation. And when the C-terminal domain is regulating the process of polyadenylation, then it has yet a third pattern of phosphorylation on its C-terminal domain. So the C-terminal domain you can think of as a loading dock, where at the beginning of the process, all of the different enzymes that are going to place this 5' cap onto the messenger RNA, they come and bind here and they work on the 5' end of the messenger RNA. Their job is done, okay? And then the pattern of phosphorylation changes and all the splicing machinery loads here on this platform, and it takes care of removing the introns and splicing the exons together. And then later on, when the process of RNA polymerization is nearing its end, the pattern of phosphorylation changes, and now all the cleavage and polyadenylation machinery comes on and takes care of making sure that the 3' end is properly processed. So here's the capping phosphorylation pattern. Here's the splicing phosphorylation pattern. Here's a movie you can go see too. And here is the the cleavage and polyadenylation pattern that happens. And here's another good video to watch that process occurring. So here's a summary of what's going on. You can take your time and read this in the book. Thanks for watching.